Today I'm going to cover some information about animal diversity. So what you should know before we get into the information about the actual animals is how they are grouped. So scientists use a classification system to put these animals into groups. Um, they assign them names as part of certain categories, and the specific animal will have two names, often referred to as the specific epithet, which consists of the genus and species name that we will get into in a moment. Some examples of that are Homo sapiens, which are humans, or Canis familiaris, which is the domestic dog. So, in the past, classification was based just on morphology, how the animal looked, but that wasn't very accurate a lot of the times because animals are pretty darn complicated. Now it's based on a mix of morphology and genetics, which gives a more accurate grouping. So, the way that scientists classify animals, there are groups that get smaller as you move down in the group. So a domain will be the largest, most general group that an organism will be in, while the species will be the most specific. A good way to remember these levels of classification, you can say that dumb King Philip came over from green Scotland, or dumb kids playing cards on freeways get squashed. So that can help you remember it because it's pretty specific and it may be one of those things that you don't want to remember, but you really need to. Another thing you need to know is that there are certain body plans and body symmetries that you see within the animal kingdom, and I'm going to go over those now. So to start, some body plans and developmental patterns we have. So um, these deal with body cavities. Acoelomates lack a body cavity. Pseudocoelomates have a fake body cavity. When you actually dissect those, it kind of looks like a tube within a tube, but it's not a real body cavity. Then you see the coelomates or eucoelomates, they have a true body cavity, which means that their body cavity is lined with a certain type of tissue, and there can be organs inside of it, and things like that. There are also certain developmental patterns that you'll see. These are called protostomes and deuterostomes. To translate directly from Latin, protostome translates to mouth first. So that means that organisms that have this type of development will develop a mouth before they develop their anus. And it's the opposite for deuterostomes. And there are only three groups that are deuterostomes. They're echinoderms, hemichordates, and chordates. We'll get into those specifics later. Then, going into symmetry, there's asymmetry, which means that there isn't really any symmetry at all. The shapes are kind of random and blobbish, like you see there. Then there's radial symmetry. So that would be the same image repeating around a fixed point, like what you see with the circle divided into slices. More specifically, there's pentaradial symmetry, which is radial symmetry involving points of 5. You'll see that in the phylum Echinodermata, which we'll also get into later. Then there's bilateral symmetry, which is two mirrored halves. You see this in humans, flatworms, many animals, pretty much anything with a head, really. And having a head and being bilaterally symmetrical actually go together. And there's an example of an image that has bilateral symmetry. So moving into the animal phyla, the first we're going to start with Porifera, which includes the sponges. These are a very primitive animal. They're the first animal and really kind of loosely qualifying as an animal. They lack true body tissues. They're mostly an aggregate of various cell types. So scientists think that they developed from groups of colonial protists, which are very simple, very small organisms that were basically just working together, and you have a sponge. Some other traits of these that they don't really have a characteristic symmetry. Some may look radially symmetrical, others may be asymmetrical. It's not really something you have to worry about. They're filter feeders, and they're characterized by a cell called choanocyte, which has a flagella and it propels water to move throughout the pores in the sponge, which helps with their feeding. And there's an image of a sponge. The next phyla is Cnidaria, which includes sea jellies, anemones, hydra, coral, things you wouldn't really expect. These are radially symmetrical, and they're also diploblastic. That means that they have 2 layers of tissue, which is kind of detailed. One of the main defining features of this group are the cells called cnidocytes, which are stinging nettle cells that can sting and stun prey. And they kind of work together with their sensory system. Like if something touches the animal, it will propel its cnidocyte and sting it and eat it. So they're predators. And you also see them alternate between body forms, a polyp and a medusa, which is basically, think about an anemone versus a jellyfish. So every cnidarian will have those 2 body forms in some way, but they're often reduced to smaller forms, and they may not be as obvious. That's a jellyfish. Next, we go into the flatworms, phylum Platyhelminthes. What you see with these is that they have bilateral symmetry and a head, and they lack a body cavity. Many are parasitic, so they have medical significance. You'll see tapeworms in this phyla, which are not good, if you didn't know that. They're parasites that can make you very sick. Then we go on to another phylum, Mollusca, which is a very large and diverse phylum that includes snails, slugs, squids, octopi, and clams. Even though they don't look alike, they still have a lot of characteristics that may just be reduced. So even though you may not see it, it may have just lessened over time. Some of those are a muscular foot, a mantle and a mantle cavity where they have their body, and a shell. You wouldn't really see a shell in an octopus. It's just reduced. They still fit into this category. And really past this point, you'll see mostly bilateral symmetry and body cavities. That's a snail. So then another group is Annelida, which includes segmented worms. This is kind of where segmentation pops up at the first time, and it's something that you see in animal phyla after this, most of them anyway. And this group includes earthworms and leeches. Leeches are parasitic, so they can hurt you, if you're in a body of water with leeches. So be careful. Then moving into arthropods, translated from Latin, this means jointed foot, and this is a very large and diverse group as well. It includes insects, crustaceans, spiders, scorpions, barnacles. There are lots of animals in this group, and many subgroups. They have a stiff exoskeleton and jointed appendages. Jointed appendages is the big one because it lets them move around a little bit, jump, grasshoppers. They can jump like that because of their jointed appendages. That's a crayfish, which lives in freshwater. People eat them. Okay, moving on, then we have the echinoderms, which are spiny pentaradial organisms that include sea stars, brittle stars, sand dollars, and sea urchins. Now these guys have a totally unique system called the water vascular system. They have a series of canals and tube feet within their bodies that helps them with movement and feeding, and no other animal phylum has this, so it's incredibly unique to this group. This is the first time that you see deuterostomes pop up. The rest of the phyla were protostomes. So this is when things start to change a bit too. That's a sea star. And then moving on, the last phylum we'll cover, because there are many phylums, there are even more than this, are the chordates. So this is also a large diverse group that has many subgroups, which I'll get into briefly. Some characteristics of this group include all chordates at some point in their life have a postanal tail, pharyngeal slits, a dorsal hollow nerve cord, and a nodal cord. This shows just a representation of some of the various parts in a chordate. Now, I'll say that humans are chordates, and we had these characteristics at some point in our development. We may have lost some along the way, like a tail. Most people have lost their tails as they develop as an embryo. So just because you don't see it doesn't mean that it doesn't have it. It just lost it some way along the way. So then moving into subphylum Vertebrata, which is a subphylum under Chordata. This includes the chordates that have a backbone. And it includes the groups, the classes out of the fish, classes Adenatha, Osteichthyes, and Chondrichthyes. So these are jawless fish, bony fish, and cartilaginous fish like sharks. Amphibians, reptiles, birds, and mammals. And then out of reptiles, birds, and mammals, these are what we call amniotes, which means that they have an amniotic egg, which is an egg where the fluid is kind of retained with a hard shell or by staying inside the mother so that the egg doesn't dry up on land. So, like I said, there are many subgroups. And here are images to represent each group. So some things to consider, finally. How much time would it take for such diverse and complex life forms to develop? Probably a long time. And then you can look at those other two questions on your own and think about the answer so that you can discuss them or ask any questions if you have them. And this is the source that I used to create this PowerPoint. That is all. That was a lot.